Is fructose metabolism happening in the chondrocyte, leading to a sudden acute rise in uric acid? Crucial to answering that question is to know whether or not the polyol pathway that converts glucose to fructose, whether or not the polyol pathway is functional in the chondrocyte. Is de novo lipogenesis activated in the chondrocyte when there is a sudden acute rise in uric acid? And if so, what are the consequences of lipogenesis in the chondrocyte? Is the downregulation of nitric oxide expected during a sudden acute rise in uric acid lead to cell death? Lastly, during the ingestion of alcohol, is alcohol found in the synovial fluid bathing the chondrocyte? And what are the consequences for alcohol exposure to the chondrocyte? The answers to these questions and more, and we're starting right now. First, remember that the chondrocyte is a specialized cell that functions in our cartilage to keep it healthy. It provides nutrients, specialized proteins, etc. We understand that the chondrocyte is a specialized glycolytic cell. In other words, it specializes in glycolysis or the catabolism of glucose. Also, the synovial fluid, and therefore the chondrocyte, which is bathed in the synovial fluid, is in a hypoxic environment, less oxygen than you find in the circulatory system. Remember that the chondrocyte has many specialized receptors. It has the receptor or the transporter for fructose, for glucose, and for uric acid. Additionally, we know that the concentration of, say, glucose or fructose or uric acid, free fatty acids, and so on, is comparable to the levels of those solutes that you find in the circulatory system. Additionally, and we're going to talk about this more, we also know that alcohol will be found in the synovial fluid at a comparable concentration to that which you find in the circulatory system. Now let's review the gout hypothesis. So in this slide, I am depicting the basic gout model. And you'll notice that in the middle of the slide that there is a membrane system, and I've labeled there's the term outside and there's the term inside. So outside refers to outside the chondrocyte, and inside refers to what's going on inside the chondrocyte. There's a couple things to remember that are fundamentally true about our model. The first is that the individual is suffering from chronic low-level systemic inflammation because of ingesting the standard American meal, which is significantly high in refined carbohydrates, unrefined carbohydrates, and so on. This results in a large bolus or a large quantity of glucose arriving at the chondrocyte, along with a large load of fructose. And if it's a dinner meal, there is the likelihood of also a large load of alcohol. The basic idea here is that the glucose enters the chondrocyte. The glycolysis pathway is completely full. We have a large pool of glucose that's sitting at the front end of that metabolic pathway. We have fructose entering the cell. We have activation of the polyol pathway that converts the glucose that's inside the chondrocyte into fructose. The fructose, which then becomes phosphorylated, causing a major decrease in ATP and phosphate inside the chondrocyte, with an acute sudden rise in uric acid. And the uric acid, therefore, is driving the derangement of the cell. So we're talking about a sudden acute rise in systemic inflammation with the transcription or the production of JNK1. We have the formation or the production of IL-1 beta. We have potentially de novo lipogenesis. We have the interruption of the nitric oxide pathway with ensuing cell death and in combination with the innate immune system, the potential for the gout flare. Now let me focus on what are the current unknowns about this hypothesis. One of the key things is we really don't know about the fructose. We know that it can be there, but is it being transported from the outside of the chondrocyte to the inside of the chondrocyte and then metabolized into uric acid? That's an open question. Secondly, we don't know for sure, or we haven't known in the past about the polyol pathway. Is it activated in the chondrocyte? Next, we know that in the liver, for example, the de novo lipogenesis, the fat switch, has been thrown, and that when we have the fructose metabolism in the liver, that we necessarily activate de novo lipogenesis. So is de novo lipogenesis going on in the chondrocyte? This is a valid question. And if lipogenesis is happening in the chondrocyte, what are the consequences to the surrounding cartilaginous area? Next, we know in the liver that the nitric oxide pathway is downregulated. The question is, in the chondrocyte, is this happening too? And because of the deficiency in nitric oxide, if this is happening in the chondrocyte, what are the consequences in terms of the inflammatory cascade, and especially to the question of cell death? Let's deal with the polyol pathway first. In this 2015 paper, Eligion and others show that the polyol pathway is indeed activated within the chondrocyte. The glucose transporters GLUT1 and 9 are upregulated. Reactive oxygen species and free radicals are also significantly increased. The nitric oxide pathway is significantly influenced. And lastly, the inflammatory cytokines underneath IL-1 beta are activated. Now the case for de novo lipogenesis. De novo lipogenesis can occur in the chondrocyte based on this 2022 paper by Park et al. ACOT1-2 is an enzyme that hydrolyzes acetyl-CoA into acetate and CoA. So when ACOT1-2 is inhibited, this causes the pool of acetyl-CoA to increase, and de novo lipogenesis is one of the consequences. This research group showed the inhibition of ACOT1-2 resulted in the upregulation of cartilage-degrading enzymes with cell death of the chondrocyte. Now for the question of nitric oxide. This slide illustrates a paper by Blanco et al. In 1995 that demonstrates overall two things about nitric oxide in chondrocytes. First, the nitric oxide system is robust. Secondly, in the presence of reactive oxygen species, specifically oxygen radicals, with the downregulation of the nitric oxide pathway, cell death of the chondrocyte resulted. Lastly, we have the question of the alcohol. In this slide, you can see that this 2015 paper by Casey et al. Showed that exposure of the chondrocyte to alcohol activated the inflammatory cascade and secondly, degraded proteoglycans. Proteoglycans are responsible for maintaining the hydration and the structural integrity of the joint. So now let's put all of this together into our gout hypothesis. Arriving at the chondrocyte, we have the deadly triad, a combination of alcohol, a large load of glucose, and a large load of fructose. The glucose comprises what we call a hyperglycemic event. If fructose enters the chondrocyte, it will be rapidly converted into a sudden acute rise in uric acid that's going to drive all of these other factors we're going to talk about in a second. Secondly, the hyperglycemia, along with the hypoxic nature of the joint and the sudden burst in uric acid, this is going to activate, we now know it will activate the polyol pathway, which will lead to the conversion of the pooled glucose into endogenous fructose, which is going to be rapidly converted into a sudden acute rise in uric acid. Lastly, remember, if the alcohol can enter the chondrocyte, then it will also stimulate the polyol pathway because it activates the first enzyme in the two-step process called aldose reductase. So glucose pool, again, is going to be converted into fructose, which then is rapidly converted into uric acid, sudden acute rise in uric acid. In this slide, I'm summarizing the process with the sudden acute rise in uric acid. Number one, and as described in previous videos, we have mitochondrial dysfunction. Secondly, we have activation of the inflammatory cascade with the activation of JNK1, which is required for the formation of the inflammasome, and also the activation of elevated levels of L1 beta. Third, and as we've learned in this video, we have activation of the de novo lipogenesis pathway with upregulation of cartilage-degrading enzymes and ultimately in cell death. Fourth, we have downregulation of the nitric oxide pathway leading to chondrocyte death. And lastly, if the alcohol does not enter the chondrocyte, we know that the presence or exposure of the chondrocyte to external alcohol leads to loss in proteoglycans and cartilaginous damage. So let's go ahead now and put this all together. Forgive me for using some notes. So basically in this slide, you can see on the left side, I have a diagram of a chondrocyte. It looks sickly because it is. And then on the right, you can see there's a membrane that cuts down through the middle of the slide. You guys have seen this before, those of you that follow my channel. And that membrane represents the membrane of a neutrophil or potentially a macrophage or one of the other innate immune cells. So in the chondrocyte, we have this perfect storm of events that we've now been talking about, where we have quantitative amounts of alcohol, glucose and fructose that have arrived at the chondrocyte. The chondrocyte already has been impacted by a chronic level of inflammatory conditions. So we already have some IL-1 beta floating around. We already have some JNK1. But now we have essentially the conspiracy of this major event, all three of these major macronutrients that are now sitting on the surface of the chondrocyte waiting to get in. We have the internalization of the glucose and also of the fructose. The glucose, because it's a hyperglycemic event, the polyol pathway converts a significant amount of it into endogenous fructose. We have the fructose, which through a series of steps produces an acute rise in uric acid that triggers these mechanisms. In essence, the cell gets sick and it begins to secrete monosodium urate, which is basically injected into the paracellular space where it can in fact crystallize. That monosodium urate is internalized into the innate immune cells like I'm showing in this diagram here. We have vesicular ATP, which is released into the paracellular space by the ailing chondrocyte. The ATP activates the P2X7 receptor, which is an ion channel, and it begins pumping calcium ions into the innate immune cell. The combination of monosodium urate, calcium, IL-1 beta, JNK1 results in the formation of the NLRP3 inflammasome. When you have all these events that come together, bam, you have the gout flare. So on this last slide, what I want to do is remind you what a gout remission eating plan it consists of. First and foremost, we have to remove the drivers of intracellular uric acid. That means we have to eliminate the alcohol. and we have to eliminate high glycemic meals, right? We don't want the hyperglycemia because we don't want to activate the polyol pathway. And we also need to eliminate fructose, which means that we need to stop eating sugar, high fructose corn syrup, agave syrups, honey, and processed food. Secondly, by making those eliminations and then cutting into the unrefined carbohydrates, we're going to effectively lower systemic inflammation. This is going to produce an environment in the cartilaginous joint which does not have chronic inflammation. By lowering inflammation, we necessarily are going to pull the concentration of JNK1, which is a protein required for formation of the inflammasome, and we also lower the production of IL-1 beta. Both JNK1 and IL-1 beta are sitting at the top of the inflammatory cascade. So when we lower them, we lower the opportunity to move towards a gout flare. Fourth, by lowering the carbs, so we do away with the sugar and the alcohol and all that, but we also are going to cut into the carbs. And when we do that, we lower insulin expression and we allow our bodies to become fat-burning machines. In the process of burning the fat, we produce ketones, and one of those ketones, beta-hydroxybutyrate, is responsible, there's solid data on this, for inhibiting directly the formation of the NLRP3 inflammasome. So I believe that the keto lifestyle is a very good option for gout remission. Give it a try and comment below. If this is the first time you've joined us, please hit the subscribe button and the bell next to it so you know when the next time is that we're publishing a video.